All right. Okay, class. So let's talk about the glial cells of the CNS. So, glial cells. We have glial cells of the CNS and we have glial cells of the PNS or peripheral nervous system. CNS, the glial cells are A, E, M, O, AMO. PNS, we have two types. We have Neurolemmocytes, and then the second type is Neurolemmocytes and satellite cells. These are the two types of glial cells of the PNS. We will focus on the glial cells of the CNS first. We will start with astrocytes. Astrocytes, so as the name suggests, they are star-shaped cells. If you look at the picture on the right hand side, the green cells, do you see the green cells? Those are the astrocytes. They form, their primary function is to form blood-brain barrier, and blood-brain barrier is actually a structure that protects the brain and prevents toxins and other harmful chemicals from actually getting into the circulation of the brain. Okay, because brain is super delicate organ and we cannot allow toxins and other buildups to actually get to the brain. So the blood-brain barrier is formed by these astrocytes that prevent all these harmful chemicals and materials from getting into the brain tissue. The astrocytes shown in the green, shown by the green cells here, the astrocytes also regulate tissue fluid composition. So it does regulate what enters and exits the brain tissue. It strengthens and organizes the tissue in the central nervous system. And if you see here, we do see a neuron, right? We do see a neuron with myelinated axon. Now let's say something happens and the neuron dies. There will be a gap, right? There will be a space, a vacant or empty space. The astrocyte will then divide mitotically to fill up the gap. Okay, so they replace damaged neurons by filling out their space. And also the astrocytes, they guide the neurons to their actual location during neuronal development. I want my students to memorize these 5 functions of astrocytes straight from the PowerPoint. So know the functions of astrocytes straight from the PowerPoint. The next type of glial cells that we will talk about is called ependymal cells. And as you see on the right hand side, the pink cube-like cuboidal cells, these are ependymal cells. These cells have cilia on their apical surface. And the ependymal cells, they line the ventricles of the brain. Ventricles are nothing but hollow cavities inside the brain. Those are lined by ependymal cells. I want you to know that brain is not a solid structure. It has some cavities inside filled with cerebrospinal fluid. And those cavities are actually lined by ependymal cells. Now the ependymal cells together with nearby blood vessels form a structure called choroid plexus. So these are I'll just highlight one ependymal cell, and this along with a nearby blood vessel, it'll form a structure called choroid plexus. And it is this choroid plexus that will secrete cerebrospinal fluid. Okay, so remember that. Ependymal cells together with nearby blood vessels form a structure called choroid plexus, which produce and secrete a cerebrospinal fluid. Okay, that's the function of ependymal cell. Remember, cerebrospinal fluid. All right, the third one is microglial cell. Very pretty, as we can see in the picture on the right. Now, microglial cells are cousins of the macrophages. Do you remember macrophages? What do? Macrophages perform phagocytosis, right? They, if they find debris or some, cellular debris or other unwanted material, macrophages just phagocytose and eat those materials, cleaning up the mess. Microglial cells, so macrophages are typically, we can compare them as garbage men. Microglial cells have the same function, but they are restricted to the central nervous system only. So microglial cells, you can think of them as the garbage men of the central nervous system, meaning they are found only in the brain and spinal cord, and they clean up any mess that they find within the central nervous system. They wander around, and if they find some mess, they perform phagocytosis to remove that cellular debris or remove part of the dead or dying cells. All right, the last type of glial cell of the central nervous system is the oligodendrocyte, and these are large and bulbous cells. If you look in the picture on the right hand side, we do see the nucleus of an oligodendrocyte. And if you look at the cell, it kind of extends a lot of arms. Do you see this? So these are all extensions of the oligodendrocyte. And what's the point? The oligodendrocyte help in the process of myelination. So this is an oligodendrocyte myelinating. So this is myelin sheath, right? Myelinating this axon. Do you see the axon? So this axon is being myelinated by the oligodendrocyte. One thing you must know, when we are thinking of the CNS, brain and spinal cord, remember one oligodendrocyte can myelinate many axons. So it's one to many. For example, I teach, let's say, 20 students in your class. So if I am an oligodendrocyte, I can myelinate 20 students. Does that make sense? So it's one to many, one to many. So that's the function of oligodendrocyte. It produces myelin sheath on the neurons of the central nervous system in one to many fashion. It's one to many. And the myelin sheath, they work as electrical insulator around the axon. So it's like a electrical tape wrapped around a wire. And it is because of the presence of myelin sheath that action potentials can travel way faster along the axon. Okay, looking at glial cells of the peripheral nervous system, two types, satellite cells and neurolemmocytes. Satellite cells, they are found surrounding the cell bodies of the sensory neurons. Right here. So what do we see here? We see the posterior root ganglion. And do we remember the definition of a ganglion? Yes. Group of cell bodies outside the central nervous system. So let's look at this box on top. We do see a spinal cord. We see portion of the spinal cord originating here. Do you see this bulb-like structure? That's called a dorsal root ganglion or posterior root ganglion. And if you zoom inside, if you blow up the ganglion, you will see inside a lot of cell bodies of sensory neurons. Remember, cell bodies of sensory neurons, they lie within the dorsal root ganglion. So group of cell bodies outside the central nervous system within the PNS, that's called a ganglion. All right. Now, if you look at each cell body, so that's a cell body right here. This cell body is surrounded by numerous pink cells. Do you see the pink cells surrounding or wrapping around the cell body of the sensory neuron? These pink cells are called satellite cells. So they are flat cells that surround the cell bodies, and they keep each cell body separated from another new cell body. They also regulate the amount of nutrients that enter the cell body. Then we have neurolemmocytes. They are also known as Schwann cells. Neurolemmocytes are the glial cells of the peripheral nervous system, and neurolemmocyte like the oligodendrocytes that we saw in the central nervous system, neurolemmocytes also help in the process of myelination. But it's one-on-one basis. So one neurolemmocyte will myelinate one axon, one-on-one basis. It's like me tutoring. So I will only tutor one student at a time. So that's how neurolemmocytes work. It's one-on-one. So it also produces myelin sheath, which works as an electrical tape around the axon. Same structure and function as oligodendrocyte, but remember it's one-on-one. Okay, so this slide talks about the difference in myelination within the CNS versus myelination in the PNS. The top picture is showing myelination in the central nervous system, where we have each oligodendrocyte extending arms and myelinating different parts of different neurons. And in case of the peripheral nervous system, shown by the picture on the bottom, we do see each neurolemmocyte myelinating only one part of an axon myelinating only one axon. And why are myelin sheaths so bright and white in color? Because they are lipid filled, so they are shiny white in color. Alright, looking into nerves, how do we define nerves? Group of axons outside the central nervous system. Remember, that's how nerves are defined. So nerves are cable-like bundle of parallel axons outside the central nervous system. They remain surrounded by 3 connective tissue wrappings. So right here on very top, you do see an axon, and the axon in this case has myelin sheath. Covering the myelin sheath, you have an areolar connective tissue covering called endoneurium. This is areolar connective tissue. Okay, group of axons are bundled together as a fascicle, as we see. So this is a fascicle. This is another fascicle. This is another fascicle, and this is another fascicle, right? So if you look at a fascicle, that remains surrounded, so this is the covering of the fascicle, that remains surrounded by perineurium, which is made up of dense irregular connective tissue. And if you look at the entire nerve, the whole big nerve, that remains surrounded by this covering, and this covering is called epineurium. So axons remain covered by endoneurium, fascicles by perineurium, and the entire nerve by epineurium. That concludes my lecture on the nervous system. So it's same thing. Okay, the end.